welcome back to genetics, and today what we're learning is actually really fun. I hope you guys enjoy today. So the first thing we're going to talk about are some special cases of genetic inheritance where we don't see a true dominant recessive inheritance type in our genetics problems, and so we have to kind of broaden the way we think about the way things are inherited. So the first one we're going to talk about is something called incomplete dominance. And so what this means is with incomplete dominance, the final phenotype is a blend or an intermediate or a compromise between the two phenotypes. So for example, we have this picture here of a red rose and a white rose. Now the heterozygote between these two traits is pink, not white, not red. When white and pink are in the same flower, when both alleles are in the same flower, they both send enough gene product to make the final flower an intermediate or a blend or a compromise between the two colors. This kind of inheritance works exactly the same through a Punnett square, but the phenotypic ratio is going to be different. So let's look and compare what happens when we have incomplete dominance compared to regular dominance and recessiveness. So in this kind of incomplete dominance, when we are giving our letters for the alleles, there is no recessive traits. There's just dominant and the other dominant. So what we're going to do is we're going to term red big R, big R. So only the homozygous big R is red. And white is big W because it's not recessive to R. So what we see then is in between these two, a third phenotype exists. And this third phenotype is big R, big W. So what we see now is when we run a Punnett square, if I cross a red flower, big R, with a white flower, and I extend my R's and W's into all of the offspring's boxes, what I find is that 100% of my offspring would be pink, or the intermediate between the two. Now, if I do a cross of two pink flowers, which is similar to doing a heterozygote cross. So if I cross my two pink flowers, RW and RW, I get a genotypic and phenotypic ratio of 1 to 2 to 1. Because my genotypes are RR to RW to WW. That also means that my phenotypic ratios are red to pink to white. So the nice part about these is there's no need for a test cross. We never have a situation where we know what it looks like, but we don't know its genotype. We always do. Because if it's red, it has to be big R, big R. And if it's pink, it has to be big RW. And if it's white, it has to be big W, big W. So all of my genotypic and phenotypic ratios are the same as each other. That is what it looks like for incomplete dominance. And remember, in incomplete dominance, we basically have this kind of push and pull, where red's trying to pull over to red and white's trying to pull over to white, and so they end up right in the middle. Another way that we can see dominance be not quite dominant and recessive is in something called CoDominance. In CoDominance, each allele is sometimes dominant and sometimes recessive. So what we mean with this is that in CoDominance, what we're going to see is sort of like a Rochambeau situation. For each individual hair on a calico cat or a roan horse, for each individual hair on the body, it either is totally brown or totally white. So the hair itself is an either or, not a middle. Okay, so in incomplete dominance, the phenotype is a middle. In codominance, it's either or, and whether you're brown or white, either or is a bit of just genetic chance. So one really famous example of this is a roan horse. So we're going to look at the coat on a roan horse right now. So this is a roan horse. It's beautiful. And what we see, and you can see it pretty clearly in this picture, is that some of the horse's hairs are white and some of the horse's hairs are brown. It's not that it has an intermediate color, that the color is somewhere between red and brown, like a light brown. No. Instead, they're either red or brown, and that's what we mean by codominance. So when we want to look at the genetics and the inheritance pattern of this, we actually start up here, and we see this bay-colored horse, okay, this brown bay horse, and then we also have a light-colored horse. And what happens is when these two horses are mated, the heterozygote has a roan coloring. So it has some brown bay colored and some palomino blonde colored hairs, and they're all scattered in between to give it this kind of roan or salt and pepper or speckled colored coat. So in a situation like this, neither one is totally dominant nor recessive. It's just that whichever one gets expressed is a little bit left to genetic chance. So what we're going to do now is we're going to once again look at what this looks like from the standpoint of the genetics. So right now we're looking at this particular example of roan coloration, and we're looking at a bay as the brown color, and small r as the white color, and the intermediate, the codominant, is the roan. So when we have the parents, big R, and small r, we would see 100% of the heterozygote, which is going to be the roan color. If then we crossed the roans, if we crossed our roans, we would see, again, a 1 to 2 to 1 genotypic and phenotypic ratio with the homozygous dominant, homozygous recessive, and the heterozygote, and also bay to roan to white. So again, we're seeing this 1 to 2 to 1 ratio. And so when we see situations of codominance or incomplete dominance, we cannot have a purebred intermediate. You cannot have a herd of roan horses that only has roan horses, nor can you have a garden of pink flowers that only has pink flowered offspring. Because those are the heterozygote, their offspring are always going to exist in that 1 to 2 to 1 ratio. A really interesting case of codominance is in our ABO blood typing. So you and I, humans, either have a blood type of A. A means that we have this A antigen present on the blood. Or you can be blood type B, and B means we have a B antigen on the blood. You can also be AB, but then very interestingly in blood type, you can also be O, which is essentially nothing, blank, empty. So what we mean by this is that if you are an O blood type, you're kind of like a ninja. O blood type can go be donated to anyone, and whether you're type A or B or AB or O, if they give you type O blood, you will not reject it. If you were blood type A, I'm A negative, if you were blood type A and I received B blood type blood, that would cause my body to have blood clots and it would cause me to die. So if I were in a car accident and I needed immediate blood transfusion in order to save my life, they would need to give me O blood, because the O blood would not cause any clots in my body, and then I wouldn't have blood clots and die. So when you are treating trauma victims, you very often use O blood because O blood is going to give you less of a reaction in more people. AB blood could receive blood from anyone because they have A and B on the surface of their cell. So if you gave them A blood, they'd be like, oh yeah, A blood, like we're friends. And if you gave them B blood, same thing, they think the same thing. And O, of course, is the ninja that comes in and automatically is secretive anyway. And so AB patients can actually receive blood from anyone. So just kind of an interesting way that blood typing works. In a genetic standpoint, there's something that we need to understand about blood. What we know is that A and B are codominant, and so we actually write them as an I, like this, with an A, or an I like this with a B. So this means type A blood, and this means type B blood. And then we have that recessive O, and so O is written as a lowercase I. So if you were going to be blood type A, you could be either AA or AO. If you were blood type B, you could be BB or BO. If you're type AB, then you have to have A and B. And if you're type O, you are two of the O alleles. So this all matters because when we do blood typing genetics, we often find parents who are, let's say, B, and they have offspring, and the offspring happens to be O, and they get mad and think that their partner has cheated, when in fact, they could be both carriers for that O trait, right? Can you see it? And they could have that O offspring. Or sometimes a parent might be AB and their partner might be B and they have an O offspring. And they wonder if that meant that there was some unequalness. Let's look at what that might look like. What if we had parents like this? Could they have a type A offspring? Let's look. We would do A and B, B and B, A and O, B and O. So this particular cross of AB with B would have type AB blood, type B blood, and type A blood are all possible. So the only blood type that couldn't come from this crossing is O. So these kinds of problems, this blood type is used in a lot of genetics problems, and it makes actually for some really fun genetics. So in tomorrow's practice, you guys will be using ABO blood typing in order to do fun genetics problems.